Hello, I'm Harrison Gabel from the Department of Neuroscience at Washington University in St. Louis, and it's my pleasure to chair this session on neurodevelopment and psychiatric aspects of TBRS. We'll have four great talks in this session covering topics ranging from molecular functions of DNMT3A in the brain to brain circuits disrupted and behavioral phenotypes that arise in TBRS. I'll kick off the session and tell you about our studies to understand the impact of DNMT3A mutations in TBRS on enzyme function and epigenetics in the brain. And then Pablo Lapunzina from Biomedical Research Network Center for Rare Diseases and Hospital La Paz Institute for Health Research will tell us about clinical findings in patients of TBRS. We'll then hear from Dr. Ana Oliveira from University of Heidelberg, who will tell us about her work on the requirement and function of enzymes encoded by DNMT3A in memory formation. And finally, we'll hear from Dr. Ching-Chun Tong from the University of Texas Health Science Center at Houston, who will present his findings identifying neurocircuit between brain feeding centers and emotional areas that co-regulate feeding behaviors and emotional modalities. This, of course, will be relevant to thinking about hyperphagia in TBRS. We'll have time for questions at the end, so please be sure to ask away. And there may be additional time at the end for extra questions as well. So I have the honor to kick off the talks and start with the first session. I'd like to thank the TBRS community for the opportunity to present our work defining the impact of DNMT3A mutations on the neuronal epigenome. So broadly, my lab is interested in understanding epigenetic mechanisms that contribute to the development of the nervous system and how these epigenetic gene regulatory mechanisms are disrupted to cause neurodevelopmental disorders. And a major focus of this work that brings us into TBRS research is our work to understand DNA methylation and its function in the brain. So typically, DNA methylation occurs at cytosines in the mammalian genome, and DNMT3A, the enzyme mutated in TBRS, is required for de novo methylation, depositing methylcytosine at CG dinucleotide in many cell types in our body. And typically, CG methylation is associated with gene repression, where a gene that's marked with methylation will tend to be off, whereas a gene that does not have CG methyl marks will be on. Now, this mechanism takes place in the neurons in our brain, but recent studies have identified a large amount of DNA methylation occurring in a non-CG context. Most of this methylation occurs in neurons at high levels at CA sites, so methyl CAs, and that's how I'll refer to it for the rest of the talk. So this suggests that the nervous system sort of has special uses for DNA methylation. And this methyl CA actually builds up to very high levels in neurons specifically. During postnatal development, you'll go from essentially having almost no methyl CA in your neurons to having it build up over the period of several months to a year in humans to very high levels, such that there's a similar number of methyl CA sites in neurons as there are methyl CG sites. And in mice, you can see a genome browser shot of analysis from the frontal cortex here, where this methyl CA is building up over a period of weeks, till there are about as many methyl CA marks as there are methyl CG marks. And we and other groups have studied how this methyl CA comes about, and we found that DNMT3A is absolutely required for the buildup of this methylation. If you knock it out postmitotically in neurons, you can completely ablate the buildup of methyl CA. And this results in severe neurological phenotypes in these mice, demonstrating the importance of DNMT3A and likely this methyl CA in the function of the brain. So this suggests that neurons have this unique epigenome that has this extra methyl mark, and that this is critical to the function of the brain. Additional evidence that methyl CA and DNA methylation are uniquely critical to the nervous system comes from studies that our group and others have done on the methylcytosine binding protein MECP2, which is mutated in the severe neurological disorder Rett syndrome. And we and other groups have found that MECP2 binds tightly to this methyl CA as well as to methyl CG, and that this binding is important for it to regulate genes. So in Rett syndrome, when you lose MECP2, you can't interpret this methyl CA, and that is likely contributing to a number of aspects of the disorder. And so we've been working to understand this pathway and how DNA methylation in the brain works. And when mutations in DNMT3A were identified in TBRS, we felt we had an opportunity to contribute to understanding the disorder by using the methods and the analysis that we had been using to understand MECP2 and Rett syndrome and the basic biology of this methylation, and applying it to understanding how the epigenome, DNA methylation in the brain, is disrupted in TBRS. And our approach to this has really been to start with the mutations first, the genetics first. So we've been interested in understanding what the mutations that occur in TBRS do to the DNMT3A protein and how that impacts the epigenome. And we've been looking both at mutations that have come out of the intellectual disability and overgrowth studies associated with TBRS, as well as some mutations that have come out of studies that start with individuals that have been diagnosed primarily with autism. We don't know if these individuals actually have TBRS and are included in this autism study because they have comorbid autism, but there are some mutations that are only in these autism studies that have been identified that we want to look at to see if there are differences between the way these two sets of mutations might affect the protein. So we've taken a sort of a three-pronged approach to understanding how mutation of DNMT3A and TBRS and potentially in an autism affects the function of the protein and what its impact is. I'll tell you about those three approaches today. One is looking at the specific mutations and how they affect DNMT3A at the molecular level. And this is in cellular and molecular assays that I'll tell you about. Then we started profiling what the consequences of DNMT3A mutation are in vivo using mouse models, looking at whether or not the mice replicate aspects of TBRS to see if they are a good model, and then to use those mice to potentially study circuits and cell biology that are disrupted and to better understand the disorder. And finally, we have this interest in this methyl CA and the MECP2 pathway, and we're interested in using that expertise and that interest to understand the consequences on the epigenome when you mutate DNMT3A. What are the molecular effects underlying the organismal level effects? And that can tell us if there's overlap between the TBRS disruption and disorders like Rett syndrome. So in the first part of the talk, I'll be telling you about what we've been doing to study mutations and what their effects on the protein are. And we're asking three questions here. First, are these missense mutations, because most of the mutations that have been identified are missense mutations, are they disruptive? It's important to determine for sure that mutations are disruptive before we think about how they might play a role in the disorder, because missense mutations are not necessarily causative. Then we want to look to say, okay, because so many missense mutations have been identified, do they have simple loss of function effects, or are there more complex effects going on? And can we understand the molecular basis of the disorder based on whether or not it's loss of function or other complex effects downstream? And then finally, we want to look at common effects across all the mutations to see what might be the drivers of neurological phenotypes in TBRS and potentially in autism-related mutations as well. So we've used a number of assays to look at how each mutation in a set of representative mutations affect the DNMT3A protein. And we've looked at cellular localization and protein expression in a heterologous neuro2A system. And then we've been able to do in vitro methyltransferase assays to look at the activity of the enzyme in a tube. And what we find with the cellular localization assays and the protein expression assays, interestingly, is that mutations in the PWWP domain seem to be affecting the localization of the protein such that it is no longer nuclear, and that the protein is lower expressed by and large. It's likely due to destabilization of the protein, and we don't know which comes first, the lack of localization or the destabilization. This domain, of course, is a domain that's important for binding with histones, particularly H3K36 methylation. So we think it's possible that by disrupting that interaction, you lose localization and stability of the protein. Now, these PWWP mutants, this truncation is slightly different, but these PWWP mutants are still active in a catalytic assay. So the rest of the protein appears to be functioning. However, the catalytic mutants show a reduction, catalytic domain mutants up here show a reduction in the activity of the enzyme in this in vitro methyltransferase assay, but no effects in the expression and localization. So we have two different ways in which you can break the DNMT3A protein, it seems, to drive TBRS Interestingly, this ADD domain, these mutations here seem to have no effect in the assays in these systems, suggesting something more subtle is going on in terms of their effects of mutation, or perhaps that they aren't pathologic. However, in the next assay I'll show you, we have evidence that they indeed are. So we wanted to dig deeper on the capacity of these DNMT3A mutations to deposit methylation, particularly across the neuronal genome. So in order to do that, we developed an assay where we could look at the capacity of the enzyme to deposit methyl CA in cultured neurons. If we plate neurons, cortical neurons in a dish, we can watch the postnatal, the postmitotic buildup of methyl CA occur in these neurons. If we do this with conditional deletion DNMT3A mice, we can use CRE to knock out the endogenous gene. And then if we add back a wild type or a mutant gene, we can ask whether or not it can rescue that activity to build up methyl CA. So mutants that disrupt the protein would be expected to not be able to build up methyl CA to the same extent that putting back a wild type protein does. And that's in fact what we see for each of the mutations we've tested, including these mutations in the ADD domain. So they all have deficits in methyl CA deposition, and the ADD mutants actually can have quite severe deficits in methyl CA deposition. And we think this may be because the ADD domain is actually a regulatory domain that interacts with the chromatin and helps to target it and activate the enzyme. However, in assays in which you're not measuring the activity of the enzyme on chromatinized DNA, you might not be able to see these effects. And so really by assaying the enzyme in neurons, we were able to detect these deficits. I also want to point out that there seem to be effects that are less severe for some mutants compared to others. And interestingly, there may be a correlation of less severe phenotypes, less intellectual disability in some of the mutations, for example, the ones shown here that have only partial inactivation of the enzyme. So this might be the beginning of a genotype-phenotype correlation. Of course, we need more data to better understand that, both on the clinical side and in the dish. So with that analysis, we've made some initial findings where the mutations we're testing are indeed deleterious and consistent with them being potentially causative for the disorder, which of course makes sense. Each of the mutations we've tested have loss of function effects on the enzyme, suggesting that a haploinsufficiency does potentially contribute to the disorder. And then finally, we're finding a common effect on CA methylation in these in vitro assays, suggesting this could be disrupted in the disorder. And because many of these missense mutations look to be null or near null in terms of their activity, and there are truncations in individuals with TBRS, we felt it was valuable to look at what haploinsufficiency of DNMT3A might look like in a mouse, starting with a simple conditional or a heterozygous constitutive deletion mouse of DNMT3A. And we can ask what the in vivo effects are of this in this model. And for example, what are the effects on physiology and behavior? Do they mirror aspects of TBRS? And then this model could be used to look at circuits and cell effects downstream in future studies, as well as molecular effects. So we started those analyses, and we have initial studies where we've looked at the heterozygous deletion of DNMT3A. And we first looked at growth phenotypes for looking for overgrowth. And we do find overgrowth of longer length of the leg bones in the animal, which would be a correlate of increased height that's associated with TBRS. In addition, we see changes in body weight, where the animals become overweight over time. This is a maturation dependent effect, which may correlate with the trends in obesity that are seen in TBRS patients as they age. So that suggests that the overgrowth phenotypes in the mice they're somewhat replicating the overgrowth phenotypes in the humans. And so we wanted to look at behavior and see if there are alterations in behavior in these mice. And we've done a number of assays. The primary effect we've seen in the mice is that they tend to have altered exploratory behavior. So if we put them in an open field assay where they are allowed to explore an open chamber, we see that they explore less and that they tend to stay out of the center of the chamber. This is often interpreted as a anxiety-related phenotype in the mice because they're afraid to be exposed in the middle of the chamber. Likewise, if we look at the animals in an elevated plus maze where they can explore either open arms, which are more anxiety producing than closed arms, they tend to stay in the closed arms and out of the open arms compared to their wild type litter mates. So this suggests an anxiety-like phenotype that may limit their exploratory behavior. We've also looked at communication and the potential for social deficits, since autism is a comorbidity in TBRS. And we do see changes in the mice and assays that are used to look at these features of behavior in mice. For example, the DNMT3A heterozygous knockout has a reduction in maternal isolation-induced ultrasonic vocalizations, which is a phenotype seen in a number of intellectual disability and autism models. And then they also show changes in their their exploration in a 3-chambered social approach assay, which is an assay typically to look at sociability of the mice. Do they like to interact with other mice? We find that they tend to just explore less in this assay rather than being less social. And we think this may be related more to an anxiety-like phenotype where they don't want to explore the open chamber rather than it being a sociability phenotype. We've also looked at learning and memory performance in these mice in a couple different assays. I'm showing you contextual and cued fear conditioning here. We don't see frank deficits in these mice that would suggest severe disruption of learning and memory. However, assaying learning and memory as it relates to intellectual disability in mice can be challenging. We do see alterations in the behavior. For example, here in conditioned fear training, after the animal is exposed to a tone and a foot shock, there is an increase in the freezing behavior, this sort of anxiety-related freezing behavior of the mice after this experience in the DNMT3A heterozygous knockout. And then in the contextual fear and the cued fear assays downstream, the mice continue to freeze more. So they're remembering the experience, but they seem to have a heightened emotional response to the experience and altered behavior. So these assays have shown us that the mice do have changes in their behavior, changes in their physiology, some of which mirror aspects of TBRS. So we think that those alterations are something that we can study in these mice, looking at what circuits might be affected and the cell biology underlying those disrupted circuits in the future. But one of the first applications we've used the mice for is to look at the epigenomic effects. So again, looking at how DNA methylation might be affected across the genome and how this impacts this MECP2-mediated gene regulation that my lab has studied, and looking to see if there's overlap in what might be happening in MECP2 disorders and what's happening in TBRS. So for this analysis, we've undertaken a number of epigenomic studies. We've done whole genome bisulfite sequencing in several different brain regions. I'll be telling you about data we have from the cerebral cortex today. And then we've been looking at histone acetylation, which is a marker and a reporter of the activity of regulatory elements across the genome. And this could tell us the consequences of changes in DNA methylation that occur. And then to look even more downstream, we can look at changes in gene expression and ask, are there changes in genes that could be driving alterations in the mice? So when we look at whole genome bisulfite sequencing data of the cortex of DNMT3A heterozygous knockout mice, we find that by and large, CG methylation is maintained across the genome. That can be seen in these genome browser tracks here, where at a very wide scale, the CG methylation is very consistent between the two genotypes. Now, if we classify different regions of the genome and ask, does the CG methylation change in those particular regions like genes or promoters or CPG islands, we don't see much in the way of effects. There's some subtle effects. And there might be a few regions that are a little bit more sensitive, but as a general rule, the CG methylation is maintained. However, if we look for focal changes in DNA methylation, searching for differentially methylated regions, we can identify several thousand DMRs, most of which are hypo-DMRs, where DNA methylation at CG sites has been lost, which is consistent with loss of the direct activity of DNMT3A on those sites or reduction of that activity. These sites tend to overlap with regulatory elements in the genome, such as enhancers over on the left here, or CPG island shores, regions right next to the promoters of genes that have been shown to be disrupted in heterozygous deletions in the hematopoietic system for DNMT3A. And these sites are sites of regulation of genes. So the changes in CG methylation we see here have the potential to disrupt gene expression patterns in the mice and contribute to alterations in the nervous system and nervous system function. Where we really see quite dramatic effects, however, is in this non-CG methylation, this methyl CA, that is specific to neurons. So we find, in contrast to CG methylation, that there's actually a global 30 to 50% reduction of methyl CA across the genome in heterozygous knockout mice. You can see that here in this genome browser track where this dotted line is at the same point on the scale. And you can see that in the heterozygous knockout, there's just a downshift of all the methylation. And this really applies across all classes of elements we've looked at for the most part, where genes and promoters and CPG and Shores all go down substantially. And we were particularly interested in seeing that enhancers, distal regulatory elements that bind transcription factors and loop to regulate promoters, they lose the CA methylation as well. And our interest in this comes from the fact that our studies of MECP2 recently have shown that one site where MECP2 seems to act is to bind at DNA methylation, both methyl CG and methyl CA, at enhancers, and to repress the activity of these enhancers. This can be measured by seeing a change in the histone acetylation. When you have MECP2 there, the histone acetylation levels will be at one level, and then when you knock out MECP2, there'll be an increase in the histone acetylation and an increase in the expression of the genes that are regulated by the enhancers. And so this is the phenotype that we've seen in the MECP2 knockout mouse. And in thinking about what's going on in the TBRS model, we're losing about a quarter of the binding sites for MECP2 across the genome, including at these enhancers. So it's possible that a subtle phenocopy of the MECP2 enhancer dysregulation could be occurring in the TBRS model mouse. So we wanted to go ahead and look at this. We looked at the methylation at enhancers across the genome, and you can see at these two example enhancers marked by K27 acetylation here, that there's a reduction in methylation. And at enhancers that are most repressed by MECP2, which tend to have high numbers of methyl CA sites, you get a very robust reduction in the methyl CA. And this, because it's 50% of a high number, is a larger number of sites that are reduced at these enhancers. And so we would expect that now that these binding sites are gone for MECP2, that the repression of the enhancer is affected, and that's in fact what we see. We see a subtle upregulation of acetylation at these MECP2 repressed enhancers compared to your typical enhancer in the genome. So that's partially phenocopying the effect of loss of MECP2, which would be losing the repression of every single methyl site in the genome. I should say that these effects are really genome-wide. We're looking at an example set of enhancers that have high methylation and then are upregulated relative to other enhancers, but all enhancers genome-wide essentially have some methylation. And we think it's actually the amount of methylation that's there before you knock out DNMT3A is going to predict how much acetyl change there is. So these are really very subtle but widespread effects on enhancer regulation genome-wide. So if the enhancer dysregulation is occurring that's similar to the MECP2 knockout, we might expect changes in gene expression that also have some overlap with Rett syndrome MECP2 models. And so we've profiled our RNA sequencing data from the DNMT3A heterozygous cerebral cortex. And consistent with what others have observed in models of neurodevelopmental disorders where there's a heterozygous loss of a gene, the changes in gene expression are quite subtle. We get a handful of significantly dysregulated genes and probably many below significance threshold effects. If we take those significantly dysregulated genes and ask, do they overlap with the effects that we see in Rett syndrome models, we do in fact see a very significant enrichment and an overlap of the genes dysregulated in the two models. So these repressed genes, these genes that go up when you knock out MECP2, they overlap with the genes that go up when you delete DNMT3A heterozygously, and vice versa. So this suggests that the gene expression effects happening in the TBRS model are, subtle partial phenocopy of the effects in the Rett syndrome model to some degree. We can also look in a sort of a non-thresholded way where we're not just demanding significance thresholds, but looking genome-wide at whether or not there are gene expression effects that are similar between the two models. And we get a very significant overlap in terms of these effects using gene set enrichment analysis and other approaches. So there is a partial phenocopy of the gene dysregulation that's going on in Rett syndrome in the TBRS models. So that's the presentation for today. In conclusion, I just want to tell you that in our analysis of DNMT3A missense mutations, we found common loss of function effects, including effects on CA methylation deposition in neurons. So we think this is going to be happening in most versions of TBRS. The heterozygous deletion model, a model of haploinsufficiency, does have defects in growth and behavioral deficits that have relevance to TBRS. So we think there is a contribution of haploinsufficiency to the disorder. We found that CA methylation is very sensitive to this heterozygous disruption, and we get a global reduction of methyl CA genome-wide in the heterozygous knockout, whereas methyl CG is affected at local sites, but in ways that could affect gene regulation. And finally, we see that there's this overlap in the dysregulation of enhancers and genes that are occurring in the DNMT3A heterozygotes that overlap with MECP2 mutants and models of Rett syndrome. So we think this means that the disorders are clearly distinct, and there are aspects of DNMT3A function that work early in development that do not overlap with MECP2. So there are going to be lots of things about the disorder that are different, the disorders that are different. But in the mature brain, we think that gene regulation in neurons, there is going to be some functionally relevant and important convergent gene disruption going on in the two disorders that would be important to think about the consequences of that and whether or not any sort of therapeutics could try to target those shared effects. And I'll just stop there, but I want to thank everybody who did the work. Of course, the most important slide. Diana Christian and Dennis Wu are two great students in the lab who spearheaded the study that I just talked to you about today, along with work from Russell Moore, Jenna Merton, Adam Clemens, and Ryan Liu, amongst other folks in the lab. And we've had great collaborations with folks at WashU and at Mizzou, including Joe Doherty, David Wozniak, Cheryl Hill, Tim Lay, Amanda Smith, and of course, Marwan Shanawi. So I'll thank you very much for your attention. I'd be happy to answer any questions.